come to the effect of mutation at biochemical level. As we know that there are some important traits, the heritable traits, they are actually related to that of some developmental processes. And in all the cases, the proteins or enzyme responsible is controlled under gene, which actually give rise to the biosynthetic processes or pathway related to the developmental process. Here I will discuss about the history, background, as well as some nutritional disorders. I will talk about the biochemical mutant. I will talk about the flower pigment formation related to the biosynthetic pathway, Drosophila eye formation, and I will discuss about the detection of biochemical mutation in different systems. Let's start. As we know that in 1909, E. Garrard have proposed the inborn errors in the metabolism in man, which is related to the biochemical mutation. Then in 1941, Biddle and Totham have found the biochemical mutation in Neurospora, as well as they have worked with Drosophila yet, and they have got Nobel Prize in 1958 for that. They have proposed the one gene, one enzyme hypothesis. That is, in a biochemical pathway, there are important specific enzymes. They are regulated by the gene function. So, from here, the mapped, cloned and gened help to elucidate the biochemical pathway by creating metabolic blocks. Mutants, especially the morphological mutants and biological mutants or biochemical mutants are found here. Morphological mutants are the outward phenotypic expression. That may be Drosophila eye color, seed color, etc. and seed size as well. In case of biochemical mutation, we can find in metabolic pathway, the enzymes, if they are getting regulated by the gene, if mutated, so they can create block in the biosynthetic or metabolic pathway causing biochemical mutation. So, biochemical mutation can be manifested at the morphological level. Now, let us see what are the morphological mutations. In case of bacteria, we can find some of the morphological mutations like auxotropes. What are they? Auxotrope means if they are plated in the minimal media lacking any one of the nutritional requirement, they will unable to grow. But if they are supplemented with that particular thing, they will able to grow. But prototropes, which are able to grow at all condition, so they will be able to grow at both supplemented and minimal media. It was found in case of humans, the albinism, phenylketonuria, as well as we can find cretinism, albinism related to the morphological mutation because it can be visible. The visible mutant means we can find in case of albinism the impairment of the melanin synthesis from tyrosine rather than hydroxyphenylalanine, and we can find in case of cretinism the thyroxine is not yet produced in proper manner from tyrosine. Now, biochemical control of gene. As we say that there is a genetic control and biochemical control. That means every enzyme is under a genetic control. So, there is a 1:1 relationship with gene and the enzyme. So, every gene control a particular specific enzyme in a biochemical pathway, and every enzyme actually drive one specific chemical reaction in the metabolic pathway, maybe more than one in sometimes. Now, inborn error of metabolism in man, it includes phenylketonuria, while the phenylalanine hydroxylase enzyme is impaired. So, from phenylalanine, hydroxyphenylalanine is not yet getting produced. We can find here phenylpyruvic acid is not turned to hydroxyphenylpyruvic acid, and phenylalanine is converted to phenyl acetic acid, which is toxic and causes the neural abnormalities. In case of alkaptonuria, it also found that homogentic acid is not converted to the acetoacetic acid, but it is converted to benzoquinone acetic acid, which is once again toxic. And in case of albinism, we can find the phenylalanine or tyrosine is not well formed towards the melanin, or hydroxyphenylalanine is not converted to the melanin production. Next one, nutritional errors in human. So we eat the fat, carbohydrate and lipid foods, and these actually help to derive energy by the biosynthetic pathway. Now during this course of biosynthetic pathway, some enzymes are there. If any problems are related to that particular enzyme, give rise to the disorders. And we can find that is not only with that particular enzyme to drive the reaction, but maybe at the regulatory protein as well as maybe related to the transporter protein. The diseases. Phenylketonuria, that is related to mental disorders, and the blackening of urine when it come in contact with the air, and that is also found in case of alkaptonuria. It was found that phenylalanine is not well metabolized and it can be converted to the phenylacetic acid, which is toxic and getting accumulated. So from phenylpyruvic acid, by the impairment of phenylalanine hydroxylase enzyme, properly hydroxyphenylpyruvic acid is not yet getting produced. It gives the mental disorders. Tay-Sachs disease. It is actually situated the gene over the chromosome 15 and related to the beta hexa gene, hexoaminidase, which destroy the GM2 ganglioside within the lysosome. So GM2 gangliosides are getting accumulated, give rise to the neurodegeneration. Next one, Wilson disease, defective copper accumulation, and it is related to the copper toxicity as well and ATP binding protein cascade, the transporter protein cascade. It is related to the transporter protein copper binding site. Again, it is related to the P-type ATPase activity and situated in chromosome 13. The next one, Gaucher disease. It is related to the glucocerebrosidase enzyme. Glucocerebrosidase enzyme break down the glucocerebroside. If they are not well broken, so fats are getting accumulated, giving jaundice, liver pain, etc. Next one, type 1 diabetes. As we know that over the chromosome number 7, there is location for insulin. Now, insulin secretion is getting signaled by different other chromosomal located genes or different loci. In chromosome 11, we can find IDDM2 genes is present, as well as we can find the GCK gene, glucokinase, regulate the glucose metabolism, and that can regulate the insulin secretion, which is responsible for type 1 diabetes, the heritable biochemical disease. Next one, Lesch-Nyhan syndrome, that is related to the very important enzyme related to the HPRT1 gene, that is hypoxanthine guanine phosphoribosyl transferase, which is needed for the destruction of the DNA RNA to the purine. If it is not well formed, giving rise to the enzyme, so ultimately the uric acid, which is not metabolized, will be accumulated and cause for the gout formation. Next one, Menkes syndrome. Menkes syndrome is related to the X chromosome like Lesch-Nyhan syndrome, and it was again the copper deficiency by intestinal absorption. When the copper absorption by the intestinal lumen is getting deficiated, it cannot support the lysyl oxidase enzyme where the copper is needed as the cofactor. So collagen needed for the bone and skin formation will not be well formed because hydroxylysine will not be formed, and ultimately muscular dystrophy happens. Next one, adrenoleukodystrophy. It is related to the long saturated chain fatty acid metabolism. If related to the transporter either or their breakdown enzyme. If it is not well breakdown, they are getting accumulated. Fats are getting accumulated within the cell. Next one, glucose galactose malabsorption. As we know that within the intestinal lumen, the galactose is broken to the glucose, and all of the glucose is transported out of the intestinal lumen by SGLT1 glucose transporter. If any problem happens over there, so they are not well transported out of intestinal lumen, causing diarrhea to happen. Next one, Niemann-Pick syndrome or Niemann-Pick disease happens over the chromosome number 18, the gene over there, which is responsible to give rise to NPC1 transporter. This NPC1 transporter is very much needed for the cholesterol traffic out of the lysosome, because cholesterol goes inside the lysosome for some modification. But if any impairment happens in NPC1 transporter, they cannot come out. So cholesterols are getting deposited by giving some disease symptoms. Next, obesity. Obesity is controlled by the chromosome number 7, OB gene. It gives rise to leptin. Leptin is a lipostat. When the fats are getting deposited over adipocyte tissues, leptins are coming out to the blood and give signal to the brain that body has enough to eat. So then, by satiety of other molecules, the obesity is getting controlled. If it is getting mutated, the uncontrolled obesity is getting happened. Next one, pancreatic cancer. Over the chromosome 18, the gene was actually mapped, because by this biochemical pathway, the mapping of the gene can also be happened by nutritional disorders. It is related to the SMAD4 gene, tumor suppressor gene, or DPC4. It can be regulated by the TGF-beta signaling cascade pathway. Next one, Prader-Willi syndrome. It is related to the misincorporation or mutation in the paternal chromosome inherited. If it is inherited by the maternal chromosome, by the same chromosome 15, it will give rise to the Angelman syndrome or disease. Here we can find the disturbance or mutation in small ribonucleoprotein N, which actually help in mRNA processing, a couple in between transcription and proper protein processing formation. Porphyria, it is related to the porphyrin ring or heme biosynthesis. There are 8 genes are there. If they are getting mutated, so they give the sun sensitivity blistering to happen. Next one, Refsum disease. This Refsum disease is related to the phytanic acid metabolism. So phytanic acid hydroxylase is getting mutated. So anyhow PTS2, this signal to send them towards the peroxisome is getting mutated. So phytanic acids are getting accumulated within the serum and blood. Next one, Tangier disease. In case of Tangier disease, once again it is related to the ABC transporter of regulating the cholesterol traffic. Once again, it helps to build up the cholesterol formation within tonsils and other organ, and it is situated over chromosome number 9. Next one, Zellweger syndrome. This Zellweger syndrome is related to the PXR1 gene, and again it is related over the peroxisome trafficking. As we know that peroxisome is related to the lipid oxidation and various other oxidation processes. So by impairment of this traffic or receptor protein, they are also getting changed very much by giving kidney abnormality and other abnormalities. Next, hemochromatosis. As we know that ferritin gives signal to secrete transferrin from out of liver in the blood. So ferritin carries iron. In this way, if HFE is getting mutated, it gives the secretion of higher amount of transferrin within the blood, which increases the iron absorption from the intestine, and on other sense, it increases the iron overload inside the body. Maple syrup urine disease, that is actually happen related to the alpha keto acid dehydrogenase enzyme complex, and here we can find it can be converted the alpha keto acids to valine, isoleucine in such amino acids, and we can find here the urine is sweet smell coming out from the children. So in case of plant like garden primrose, we can find that these anthocyanin pigment differs. Why? Because somehow the anthocyanin pigments are redder and somehow they are bluer. Now bluer can be found if they are anthocyanin pigments are attached to the sugar moieties or hydroxyl moieties. If the enzymes are present controlled by the genes, they will be bluer. But in some cases anthocyanin pigments are redder. Why they are redder? Because these all the cases the sugars or hydroxyl groups are methylated. Next one, very interesting, eye color in case of insect like Drosophila. So what is going on? Insect eye pigment. We can find in case of Drosophila different series of eye color. So we can have vermilion, cinnabar, garnet, sepia, different shades of brown and red colored eye phenotype at the phenotypic observation. So that is due to the presence or absence of some pigment. Some are bright redder like vermilion, or some are bright brown the disappearance of the red color. So brown is found in case of garnet and sepia. So that is due to some biochemical effect. What sort of biochemical effect? As we find that in case of Drosophila, two pigments are produced, drosopterin and omnochrome, by two different biosynthetic pathways. After synthesis of the pigments, they are getting accumulated or with the protein, and they are precipitated over the eye disc to give rise to the coloration. If one pathway is getting mutated, other pathway predominates to give a specific color. As we know that the drosopterin mutation pathway happens to give the redder color, but if mutation happen in the omnochrome pathway, give the brown color. Now proposed result. So there is a pathway. Now before going into the pathway, we have to know how this pathway was developed. So from tryptophan, actually the chi-urenine is getting produced. It was observed in case of cinnabar mutants that the block was gone if the chi-neurin is given. Now next one, cinnabar. Cinnabar is due to the block in the hydroxylase enzyme. The hydroxylase chi-neurenine hydroxylase. So from chi-neurenine, the 3-hydroxy-chi-neurenine is not properly produced. That is actually happened, but no mutation or no mutants color are found related to the formylase, but can also be found in case of cardinal mutation color due to the synthetase gene. So let's see what is the biochemical pathway developed by these sort of blocks. So this is the pathway. So from tryptophan, the chi-urenine is getting produced, and from chi-urenine, ultimately the xanthomterin, this particular omnochrome is getting produced. So this is the omnochrome pathway. Next one. Actually, this was proved by eye transplantation in Drosophila. It may be autonomous or not non-autonomous eye transplantation. First, it was found that in case of wild type, if vermilion or cinnabar is given, then wild type will be produced, because from the wild type subdiffusible enzyme can give up the metabolic block to generate the wild type. And then again the reciprocal transplantation, that means if not within the wild type the vermilion or cinnabar is given, that is within the vermilion or cinnabar if wild type is given, then from the wild type once again the diffusible enzyme can come and give up the metabolic block to generate the wild type. Interesting thing, in vermilion if cinnabar is given, then again it will help to develop the wild type. But within the cinnabar, if vermilion is given mutant, then it will not be able to cause the wild type to generate. That helped to know that vermilion gene blocking is coming before the cinnabar. So from this we can totally elucidate the biochemical pathway, that is vermilion gene related to the pyruvase, then cinnabar related to the hydroxylase, ultimately giving rise to xanthomterin on omnochrome synthesis. Next, and we can also have the proposed pathway for drosopterin synthesis. Now let's talk about the biochemical mutation in Neurospora. Neurospora crassa. It was found that detection was found by Biddle and Totem in auxotrop within the minimal medium, but in the supplemented medium it was found by given specific pyridoxine, thiamine, it will able to grow. What was found? Total isolation method. So within the ascar 8 ascospores are grown and segregation are well developed. First it was grown in the complete medium, then it will be transferred to the minimal medium, and if it is monitored only grown in the total medium or complete medium but not grown in the minimal medium lacking some of the nutrition requiring substrates, then it will be the auxotrop. If grow on both the medium, they are the prototrop. This can also be done with the complementation test. Why? In case of diploid heterozygote, the segregation happens. If mutants happen in same chromosome, they will fail to complement. So auxotrop will grow. But if we have the prototrop, so that means mutant on the separate chromosome, they will complement each other. In Salmonella Typhimurium, we can find severin tryptophan mutant. Like in case of TRP8, it will only grow when anthranic acid, indole, indole glycerol phosphate as well as tryptophan will be given. In case of TRP2 and 4, it will not able to grow by giving any other things like precursor as anthranic acid. It will able to grow when indole, indole glycerol phosphate and tryptophan will be given. Next, in case of tryptophan TRP3, it will able to grow when indole and tryptophan will be given, and in case of TRP1, 6, 9, 10, 11, it will able to grow when tryptophan will be given. So, from these, from chorismic acid, ultimately how by indole glycerol phosphate indole, ultimately how tryptophan can be synthesized, it was mapped. In case of E. coli, there are A, B, C, D, E. 5 mutants are found, which is also almost related as Salmonella Typhimurium related to the tryptophan synthesis. Histidine metabolism, in case of Salmonella, it was mapped 500 mutants, and it is related to A to H loci and help to elucidate the pathway. The mutation frequency with according to the Poisson distribution ratio. If mutation rate per division is mu, then mutation event will be mu n. Then the frequency by Poisson distribution ratio F0 will be e to the power minus mu n. Now, detection in the biochemical mutation in different systems will include first in Drosophila. The sex-linked mutation by altered inherited phenotype will help to also elucidate the autosomal recessive genes. And detection in human was found electrophoretically with different types of isozyme. Slow and fast moving glucose 6-phosphate dehydrogenases was found. Null mutation gives the absence in the electrophoresis. So, abrupt change, deletion within the protein will vanish the protein from the electrophoretic gel. So, it will help to detect the biochemical mutation. Detection in Neurospora, that I have already said that the phenotypical change can be observed by auxotrope as well as prototrope. So, auxotrope will unable to grow in the minimal media, can only be grown in the supplemented media with that specific requirement. So, it will help to grow in the histidine medium as well. In bacteria, by Liederberg and Zindler, it was found the antibiotic resistant mutant to grow by the presence of streptomycin, as well as the phage resistance help to grow to count the number of mutant cell as well. So, mutation rate 10 to the power minus 8 can easily be counted from the antibiotic or phage resistant plate. In virus, so the conditional temperature sensitive mutation in bacteria was found, as well as conditional lethal, the suppressor sensitive mutation were found by viruses with differential types of plaque formation. It was found in case of virus, the differential plaque sizes with respect to tobacco mosaic virus was found, and the host range can be increased with the mutation in case of virus as well. Next, in rodents, it was found in case of Chinese hamster cell when purine analog 8 as a guanine is given. So, the auxotropes will, the mutant will easily can be screened to grow over there. Not only that, with the mutation, the different types of phenotype can be observed in mice like monoagouti, like chinchilla, like piebald, dilute, pink color eyes, pink eye, etc. So, I have discussed about the biochemical mutation related to the nutritional disorder, and it will actually help to elucidate the anabolic pathway in flower and Drosophila, as well as I have detected the biochemical mutation in different system, the analysis system as well. Thank you.